Hello, and thanks for joining us today for the latest Technology Networks webinar, The Future of Preclinical Drug Research and Introduction to Organ-on-Chip. I'm Holly Large, Editorial Assistant for Technology Networks, and I am here to moderate the event. I'm pleased to have Vaish Manoharan joining us today as your presenter. A warm welcome to you. Graduating with a biomedical science degree at Keele University and holding a master's in immunology from Imperial College London, Vaish joined CN-Bio in 2019. She has over 9 years of cell culture experience, including working with 3D cell culture. Following the webinar, we will have a Q&A session, and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the left-hand side of your screen and click Submit. We will answer as many questions as possible during the time available. For any questions we don't have time to get to, we will be sure you are contacted offline with an answer. If you experience any technical issues, please click the question mark in the top right-hand corner to request support. Please remember, you can ask questions at any time, even whilst watching the webinar on demand. So, without further ado, I will now hand over to Vaish.
Thank you, Holly, for the introductions. Today, I will discuss why there has been an increased interest in organ-on-chip systems in the recent years. In the recent years, organ-on-chip term has come into common use. Others in the community may use microphysiological systems. For simplicity, in this presentation, I will be using organ-on-chip. The two terms are interchangeable. I will then discuss how do OOC systems fit within the pharmaceutical industry. I will present CN-Bio organ-on-chip system and highlight the application of the drug discovery process. So what is an organ-on-chip system? An organ-on-chip system is an advanced in vitro model, typically featuring 3D cultures of human cells with fluid flow. The goal is to grow cells in condition which match the human body, so they will respond to drug and other chemicals in the same way as a human would. The OOC community. The OOC community has matured over the past decade with the support of academic research, a number of spin-out companies, and significant engagement within the pharma industry. There are different organizations and consortiums to help raise and promote the use of OOC systems. As an example, the Orchid Consortium. The goal of Orchid Consortium is to create a roadmap for OOC technology. This identifies potential roadblocks and solutions, raise awareness, and to implement OOCs in science, R&D, and regulatory guidelines in Europe and elsewhere. Academics have developed and published on many innovative OOC systems. We are seeing OOC companies taking these systems and developing them towards commercial products and service offerings. An ecosystem has begun to grow with industrial partners supporting development through technological expertise, financing, and pharma companies accessing technology through collaborations. We are starting to see OOC systems being adapted and utilized on active drug discovery programs. CN-Bio has been part of this community for a decade with strong links to academia, biotech, and pharma companies. The rise in publications illustrates the growing interest in the field. This has been mirrored by the founding of numerous OOC startups such as CN-Bio. We are now seeing publications from across academia, OOC vendors, and the pharma industry. CN-Bio is contributed to this growing evidence-based paper. A paper has been accepted for publication with the University of Cambridge on our NASH model and a paper in preparation with the FDA on the evaluation of microphysiological systems. So why has there been an increased interest? The biggest reason is that 90% of drugs which enter clinical trial fail to become approved medicines, as current preclinical methods such as conventional methods, 2D cell culture, and animal models are not always effective tools to predict drug safety and efficacy. Standard methods, such as 2D monolayer cell cultures, which are overfed and do not represent physiological characteristics of an organ, cell type, or tissue. There are many examples in which mouse and other animal models have been shown to be poor predictors of the response of human to new medicines. This lack of translatability between animal and human data is a key challenge for the pharma industry. Due to these issues, millions of pounds are wasted each year on the development of medicines which fail in the clinic. To cut the cost of drug development, it's important to improve the predictivity of preclinical screening to enable a fail early, fail cheaply scenario, eliminating ineffective drug candidates as early as possible. So the question is, how can OOC systems be placed within the drug discovery process? OOC has applications throughout the drug discovery and development process, starting from the target discovery all the way through to supporting clinical development. The CN-Bio system has enabled these applications right across the process. For example, OOC models can be used for disease modeling during early discovery phases, identifying novel drug targets and understanding the mechanisms underlying disease processes. These same disease models could additionally be used to support clinical development and inform clinical trial design. At CN-Bio, we have developed advanced models of HBV and metabolic liver disease, which have been used by researchers to advance compounds through the development process. Within DMPK, CN-Bio's OOC system has been used to determine the metabolism of compounds and, in the future, multi-organ systems with inter-organ crosstalk such as gut-liver model will be used for great levels of translation. At CN-Bio, we have a gut-liver model coming out soon, the TL6, which I will discuss in further detail later on. So why are we doing this? Looking for the future, OOC has the potential to create a patient on a chip, enabling precision medicine with the response of an individual being predicted and treatments recommended. These systems will contain multiple microtissue structures representing various key organs, all with the same genetic background and fluidically linked together, mimicking the flow of blood around the human body. Now, I want to talk about our organ-on-chip system, the CN-Bio PhysioMimix. The system was born out of a technological race sponsored by DARPA between MIT and Harvard. The PhysioMimix system has been developed over numerous years and became commercially available 2 years ago. The platform has been implemented into 8 of the 20 pharma. We have also collaborated with leading academic partners, including Imperial College London, and more recently with the Centre for Drug Evaluation and Research at the FDA. Just a note on this point, within this webinar series, there will be a webinar presented by the FDA on the strategies to evaluate MPS systems for drug development on the 15th of July. CN-Bio is developing a second instrument based on IP licensed from Vanderbilt University, which enable the accurate modeling of pharmacokinetics and drug dosing regimens in vitro. As you can see, the simplicity of the PhysioMimix platform is in its design. The overall system includes a controller specifically designed to provide accurate microfluidic flow within the OOC plates. The docking station supports multiple types of OOC plates and sits directly inside your incubator to maintain consistent cell conditions during your experiment. 2 docking stations can be added to increase throughput as required, a unique feature of our system. Our existing customers have commented on the ease of use and simplicity of setup, therefore making it easy to transition from standard 2D cell culture to 3D microtissues. We currently offer 2 different consumable OOC plates. All plate types have an open well design, allowing simple access to cells and media throughout your experiments. The T12 is used to create a multi-cell type barrier model, for example, the gut, lung, or skin. It can then be used to model drug transport, toxicity, or disease mechanisms in these tissues. The LC12 is designed specifically for culture of liver cells and backed by extensive research from CN-Bio and MIT. The LC12 plate. Each plate has 12 independent wells with micropumps that allow a tunable flow rate. It is PDMS-free, so there is low non-specific binding, where CN-Bio uses chemically inert COC, commonly used in cell culture plasticware. Our plates come with pre-coated 3D scaffolds in each well that are specifically designed for the culture of multiple liver cell types. The T12. Each plate, again, contains 12 independent wells to create barrier models using standard transwells. The micropumps provides flow to the basolateral side of the transwells throughout the culture period. The plate is flexible to accept pre-formed transwell models or tissue models that can be developed under the presence of flow in the platform. Now let's look at the fluidics, which is the heart of the system. When you seed your cells in the plate, the controller automatically directs the flow down into the scaffold to allow the cells to form the 3D structures over the first few hours of culture. The flow then continues to run throughout the experiment through the scaffold to provide biomechanical stimulus, oxygen, and nutrients to the cells. For the T12, flow on the basolateral side of the transwell promotes barrier models with more pronounced 3D topology. For example, we have seen in our gut model, flow acts to increase permeability, bringing the model closer to physiological parameters and therefore providing greater levels of translation. On both plate types, media samples can be simply taken from each well, and the 3D scaffolds or transwells can be removed to allow analysis of the tissue structures. How does your typical experimental design work? Let me give an example using our LC12 liver model. You start with your cells of your interest. These could be your primary cells, cells originated from stem cells, or a more simplistic cell line, and seed them into the plates. From day 4 onwards, a disease state can be induced. So, for example, introduction of a viral infection. And at the appropriate time, drug dosing can be initiated. We aim to dose compounds in a manner that closely represents the dosing regimens performed in patients. We have successfully used a wide range of modalities in the platform. Cells are typically cultured for a number of weeks and remain viable throughout, well beyond the classic cell culture methods. After your experiment, data is generated using the media or tissue, so you can perform a wide range of standard endpoint analysis, for example, omics, microscopy, histology, and biomarkers. You can obtain 1 ml of media to run various biomarker analysis and large-scale tissue on scaffolds to extract genetic material. Our platform has been adopted and utilized in numerous applications within the pharma drug discovery and development process. The CN-Bio system falls in nicely as part of this roadmap. So, for example, our current customers are using these applications in disease modeling to study NASH, HBV, breast cancer metastasis in the liver. We have our customers testing in toxicology to investigate the DNA damage. And within DMPK, we have customers testing the human-specific metabolic profile of large and small molecules. As an innovative company, we always strive to develop new products to help research with the organ-on-chip community. Here, we have some products coming soon. The TL6 is a true two-organ plate where the flow rate will be adaptable for each organ to fully represent the microphysiology of the target organ. We'll be implementing a unique sensing solution to support real-time monitoring of the cell health and oxygen levels using biosensors. This will provide an efficient mechanism for cell culture QC and provide valuable insights into cell viability. Just to recap on the main points I have discussed today, OOC systems are becoming essential to support efficient drug development. CN-Bio systems are adaptable, user-friendly, and cost-effective. Regulators are now establishing strategies to evaluate OOC systems. Pharma, biotech, and academia are rapidly adopting the CN-Bio system. Thank you so much for listening. We have a series of webinars coming up. The next webinar will be presented by Dr. Michaeli Racker from the University of Cambridge, who will be discussing 3D NASH models in our OOC system. I will be happy to take any questions now.
Thank you very much, Vaish. I'm sure you'll agree that there's some excellent points raised during the presentation. We'll now begin the Q&A session. To submit a question to our speakers, you should enter your question into the box on the left-hand side of your screen and click Submit. We will answer as many questions as possible during the time available. We've had several questions submitted already, so I'll move straight on to these. So the first question we have is, you mentioned you don't use PDMS. Why is this important?
Hi, so PDMS is known to be highly binding to chemicals and drugs when assessing drugs PK/PD. There is a need for the most inert, low binding components in the system. We use COC, which is currently known as the most inert material available, and it does not actually impact on the cell growth, but impacts on the drug.
Okay, fab. Thank you for answering that one, Vaish. So the next question we have is, you mentioned that your system is easy to use. Can you explain this further?
So our system is user-friendly, flexible, and it can be used for various cell formats such as cell lines, primary cells, commercial inserts in our system. And with our system, we can also do long-term cell culture for over 4 weeks. And with our endpoint analysis, as mentioned in the presentation, you'll be able to do large-scale sampling for more than one endpoint analysis.
Okay, fab, thank you. The next question we have is, why don't you use PDMS for LC12 and T12? Is the biocompatible polymer used to assist the cell growth?
So I vaguely answered that question before. So both LC12 and T12 are actually PDMS free. So with that, it means that there's low non-specific binding.
That's great. Thank you very much, Vaish. The next question, you discussed a number of applications in your presentation. Are you looking at NASH?
Yes, we have. We actually have an in vitro 3D microfluidic model using triple culture of primary human hepatocytes, compass cells, as well as stellate cells. And these are treated with a specialized fat-loading media to be able to recreate a NASH phenotype. The model can actually be closely replicated to key disease markers and allows for drug testing to be performed in like a human relevant model over a period of weeks. And we actually also have a NASH fee-for-service in-house, where, for example, if you had like a compound of interest or drug of interest, you would send it to us and we would be able to test it in-house for you using our NASH model and be able to get results back to you within 6 to 8 weeks, depending on how big your research project is. And we actually have a webinar as well next week from Dr. Michaeli Racker, who will be presenting more on our NASH model with the OOC system as well.
Okay, fab, that's great to know. Thank you very much, Vaish. The next question is, can OOC results be submitted to regulatory authorities for IND filing?
Yes, they can. All regulators have said that they would encourage IND submissions with OOC data, and OOC data can be used for both efficacy and data tox packages in IND.
Thank you very much. The next question, what has been done with heart cells? What aspects of cardiac function can be demonstrated?
So with a heart cardiac tissue, it can be modeled in OOC platforms. We've actually used iPS cardiomyocytes to model cardiac tissue, and this has been linked to models of the liver to study cardiac liver toxic crosstalk.
That's really interesting. Thank you very much. The next question is, what are the research initiatives at CN-Bio for skin models? What has been done or achieved so far?
So for skin, you can actually use the transworld model, so that's our T12, and these can be generated from numerous sources, including pre-formed tissue. They can then be put into the OOC pattern to be put under perfusion or linked to other organs such as liver or lungs, basically your organ of interest, and then this can be used for numerous amount of applications, for example, tox, cell health, or drug absorption.
Fab, thank you very much, Vaish. The next question, could this system be used in other species, say feline or canine?
So we've actually not specifically done with other animals, but it's not to say that we can't actually try it with different animal models in our system. We have tested it on rats, but in terms of canine, we've not specifically tested that.
Thanks, Vaish. The next question is, you mentioned that regulators are evaluating the OOC for drug applications. Could you tell us any more about that? Any examples that you could share?
So actually, in this webinar series, we have a webinar with the FDA who will be evaluating the strategies of the MPS system. So I'd really recommend that you listen into that. We also have a paper coming out soon with the collaborations with the FDA that we're able to, once the paper does come out, that we'll definitely be able to share with our participants.
Brilliant, thanks Vaish. The next question is, how much is the possible sample volume per each well?
So for each well, you can get about 1 ml, which again reinforces the point that you do get quite a bit of volume to be able to do a lot of endpoint analysis, and that's 1 ml per well. And with our plates, you do get 12 independent wells.
Thank you. The next question is, can the system be used technically for any cell line? So, for example, cancer cells, non-mammalian, etc.
Yes, we've tried a various different types of cell lines, so it's not to say that you can't try with different cell lines. It is varied in terms of our platform and adaptable for different types of cell lines.
That's great. Thank you very much, Vaish. That's all we have time for today. Just to remind everybody, any questions that we didn't have time to get to will be answered offline as soon as possible. You can continue to ask questions even when watching this webinar on demand. The webinar will now be made available on demand. If you have any friends or colleagues that you think may be interested in this webinar or any future events, please feel free to share the link. Thanks again for your time.